It's a pleasure to be here this morning. So far today you've had two talks. One has been more basic biology, and the last one, fascinating discussion of technology and how it can be used to understand something about biology. My talk's going to fit somewhere in between those two. I'm going to tell you a little bit more about a specific biological system, in particular the cytoskeleton and how it's used for motility. And I'm going to try and talk about the ways in which the mechanics of the environment are important for the functioning of that biological system. So rather than the discussion you just heard about how mechanics or shape change is to be avoided, biological systems use that information. And I want to show you a couple examples of where that is both good and where that is bad. I'm also going to talk to you about a couple different ways of gathering information, of sensing biological systems. In particular, I'll talk about optical methods and force microscopy methods for gathering information about how biological systems respond to their environment. So the picture I have up here is of a fish keratocyte cell. So if any of you have had a goldfish before, if you were to pluck a scale off of that goldfish with proper approval and culture that in a dish, off would crawl a sheet of cells composed of these individual cells. Eventually those individual cells would crawl away. And those are wound healing cells that participate in patching up the fish now that you've damaged the scale. And so these cells have been used as a model system for understanding many of the features of motility, of crawling motility. So I'll tell you a little bit more about crawling motility. What you're looking at inside of the cell is some fluorescently labeled proteins. So you heard this morning about actin. So in red is actin, and that's a set of polymers that are involved in pushing out the leading edge of this crawling cell. What you're looking at in green is a protein called vinculin, which is involved in attaching to the substrate. So the reason why this cell doesn't get washed away in fluid or on the outside of the cells, it's able to hold on. And then in blue is the nucleus. And for the kinds of things I'll be talking about today are the very short timescale processes. The nucleus is really kind of dead weight. Now the nucleus, of course, is important for the ability of the cell to maintain itself. But for many of the processes that I want to describe today, you don't need the nucleus. The nucleus is there as a reference, but all of the behaviors that I want to talk about really rely on what proteins are present and their ability to interact, sense, and respond on their own. And you can actually dramatically show that you don't need the nucleus by slicing it off of these cells, and the cells still crawl. So the questions that my lab is interested in are twofold. How do cells respond to their physical environment? What are the signals that are interpreted? How are they interpreted on the molecular scale? And what sort of responses do cells undergo? And then secondly, how can we rebuild these processes? Are there ways of generating both new understanding about how cells are organized and how they're built, as well as creating new sensors? Is it possible to take some of what's known about individual molecules, about how they're organized, put them back together, and use them in some engineering way? So I'll talk a little bit about the second, but mostly about the first. So the outline for today involves a few different things. First, I want to motivate why we're studying this process. Second, I'll tell you a little bit about some of the tools that we use for this, in particular force microscopy tools. And then I'll show you three different examples of where mechanics plays a role in biology. In the first case, leukemia, this is a situation in which mechanics is playing a negative role. There's a problem associated with leukemia that is due to changes in mechanics. Platelets, which are critical for a range of different repair mechanisms, I'll talk a little bit about how they are able to sense things about their physical environment. And finally, I'll go inside of the cell and talk about actin networks and how those isolated networks are able to respond to forces as well. And finally, I'll end with a discussion of some of the tools that we're working on to allow us to reassemble more complex systems. Okay, so let me start with some movies. So my background was in engineering and applied physics, and when I was towards the end of my PhD, working on developing microscopy techniques, I became enamored with motility, movements of cells. I think it's worth remembering as you look at a swimming cell like this organism, that on this scale, so that disc is about the central part of the cell is maybe 6 to 8 microns in size. And there's no amount of engineering that we can do that can reproduce the kinds of motions that cells create. And so they really represent a remarkable engineering challenge, both for us to understand how they work, and then a challenge for fabrication. Can we actually put systems together, either out of biomolecules or other materials, that would let us get these sorts of behaviors? And so that's what really attracted me to movement, was the fact that here is an example of a microsystem that is able to do dramatic things, and we really don't understand how to do that. Granted, biology's had quite a bit of a head start on us, but still, we should be able to understand molecularly what's going on. Does anybody recognize this organism? You probably don't run across it on a daily basis, but if you've ever been hiking in the mountains and you're told not to drink the water because it might have something in it, that's sometimes what's in it. It's not in this form, it's in a cyst form, so this is Giardia lamblia. And if you feel really sick after going hiking, this may be the cause of it. So these beautiful organisms colonize your small intestine, and they're able to hold on. And actually, the mechanism of attachment is a subject of debate still. But they're able to swim around, and they're able to move thanks to these flagella that are flapping around. So in some parts of the body, infectious agents are able to swim. Here's another infectious agent, and this is called spiroplasma. Hopefully you haven't run across this one. This one really is nothing to fear. This is if you're a honeybee or a plant, you would be a citrus plant, you'd be very afraid of this. But other than that, these spiroplasma, these are molecules. They don't infect humans, so there's nothing to be afraid of. But they have a remarkable ability to move. They have no external appendage. You saw that flapping tail on the Giardia. Those flagella are able to move the Giardia around. In the case of spiroplasma, there's no external appendage that allows it to move. And so in studying this organism, what we found was it has a helical shape, and what it does is it changes the helicity of its body, and it propagates that down in the form of a wave, and then starts at the head and switches the helicity of the body back, propagates it down. So it's able to swim through the viscous material without an external appendage. It essentially uses its body as an external appendage.
And how big is this?
So this is 3 or 4 microns long and about 200 nanometers wide. So a really, really small organism. One of the simplest ones out there. So here's an organism to be afraid of. Again, one that you can get infected by. If you go and have dinner and you have undercooked chicken or other products that haven't been heated high enough temperature, you could get Listeria monocytogenes. This is a particularly nasty bug, or if you have really soft cheeses as well. That's in France, it's expected that a certain number of people will die from Listeria monocytogenes because they don't want to have to pasteurize all the cheese. So this is a remarkable bacteria, and it also doesn't have an external appendage, but it's able to move. And it's able to evade the immune system so that your body doesn't even know it's there. And the way it does this is by getting inside of cells and then moving around as a result of its ability to hijack some of the host cell proteins and move. And in fact, the proteins that it hijacks, the major one, is actin. So it's able to redo what cells are doing when they crawl. It's able to move itself around with that process. Is there a question?
So what's the cytoskeleton element involved in spiroplasma movement? Is it
good question. It's not clear yet. So there are a number of fibril proteins that are present, but exactly what the mechanism is that's causing this switch, we don't know. Good question. So Listeria monocytogenes, we do know, and there are a number of other bacteria that also hijack the host cell cytoplasm with proteins in it to be able to move around. So the kind of motility that I want to tell you about is not the swimming kinds. It's rather the crawling kind, because in most parts of the body, you don't really have a lot of space to swim, but there is space to crawl. And so let me show you what that fish keratocyte is able to do. So again, this is the cell that I showed you in the very first picture. And if you do observe it over time, so this is about a 5-minute movie, it's able to glide across a glass cover slip. So this is like moving across a relatively flat scale as it tries to find the fish and patch it up. And it has this beautiful shape, and it's able to maintain its movement in a very highly directed way over long periods of time. Now all this process is taking place over 5 minutes. So what sort of thinking do you think this cell is doing? If thinking is even the right word. It has to process information about its environment. It has to know something about, am I going in the right way? Is this the best decision or not? So it's having to rely on chemical sensing. And what I want to show you here is physical sensing as well. So here's a slightly different situation for this same cell. So it's about to run and hit this barrier. So this is a little line at the top. It's really about a 5-micron step. And so it's crawling and doesn't have eyes, of course, so it doesn't know that this step is ahead of it. So how many people think that when it hits this step, it's just going to stop? So that's option number 1. Number 2 is that it's going to crawl right over the step. Doesn't care about a step. Number 3 is that it's going to bounce off like a ball hitting a wall. So how many people think it's just going to give up? Okay, good. So you have some faith in biology, you have some faith that cells will continue to process. How many people think it's going to crawl up and over?
How big is the cell?
So the cell is probably 20 microns across, and the height is under a micron. So you think people think it's going to cross over?
Yeah.
And how many people think it's going to bounce off? All right. Okay. So let's see who's right. And if you're right, you can leave any time. So you already you can go to lunch early. So it's hitting, smashed into it, and it bounced off. So bouncing off, people are right on. So in order for it to do that, it's had to completely reorganize over the course of about 5 minutes. It's the organization of its cytoskeleton, the internal structure that is allowing it to generate force, that's allowing it to hold on to the surface and allowing it to move. And you'll notice as soon as it hits, you begin to see some ruffling there at the edge. So the fact that it's hit one place is showing up in another place. So there is some sort of signal transduction, there's some sort of transfer of information about that contact to distal location.
So then,
Yep.
if the barrier is of the same height as the cell, then, well, we know that if the barrier is very little, maybe a few nanometers, certainly it will crawl.
It'll go right over. Yep. Mm-hmm.
So
If it's closer to the height of the cell, actually the cell extension at the very leading edge is around 300 nanometers. It's quite thin. So if the height is around that, it'll go up and over. It's larger barriers like that. When it realizes I can't get around that, I'm just going to bounce off of it. Now, immune cells like neutrophils or macrophages don't have as flat a protrusion, and so they're able to go over much more complex geometry.
What's the barrier?
So this is just a step in a PDMS substrate. So the point of me showing you this was to point out that the very process of crawling has to have built into it some ability for the cell to sense, some ability for the cell to recognize that it's not going to be able to go forward, and it has to somehow reorganize. And the question that we wanted to tackle was, what exactly are the molecular mechanisms that allow for this sort of reorganization?
do I also see some deformation of the PDMS barrier?
This has been, there's a little bit of an overhang, I think, and so you see a little bit of the cell either going under or over that, but the bulk of the cell does not make it over the ledge. Okay, so I'll show you one more and perhaps the most dramatic example. Question?
Does the stiffness of the barrier affect whether or not it'll go over?
That's an interesting question. I expect that it would, because the mechanism that we think is at work is an ability for this cell to sense whether or not it can push. And so if it can push, then it's more likely to continue forward. But we haven't exhaustively tested that. So this last example is one that shows a coordinated movement. So this is a blood vessel of a fish. So this is a zebrafish, which is a model organism that many people use to study wound healing. And what you see at the top there is a puncture wound. So someone just took a needle and poked a hole into this zebrafish. And the question is, what is the body's response to that? What is this organism's response to that? And what you see on the lower part of that image is this blood vessel, and from this blood vessel come white blood cells. And so they are also crawling, and they're recognizing that there's a wound in that location, thanks to the presence of bacteria that have entered as that needle was poked into the fish. And so that chemical signal is being smelled by these cells, and these cells are moving towards that location. Now remember that these are about 10 micron in diameter bags, and they're filled with enough molecules and enough sense to be able to figure out which way to go, to traverse a very complex physical and mechanical environment, and end up there. And they don't just sit there. Once they're there, they participate in the process of clearing the wound of invading bacteria and other organisms. So these are highly complex and highly capable structures. And the question is, how really do they do this? How do they traverse their mechanical microenvironment? Yeah?
So is this a section, or how exactly does this image go?
So this is not a section. This is the fin of the zebrafish is very, very thin. And so you can actually do light microscopy through it. You'll notice, for those of you who have done microscopy, you'll notice that there's a bit of a shadow. Kind of has that three-dimensional look. And that's characteristic of differential interference contrast microscopy. Yep.
How long does it movie take?
I don't remember how long this is, but macrophages or neutrophils crawl something on the order of a micron per minute. So this is sped up maybe 20 to 30 times, something like that. Yeah.
I still have one question for the cell. Do you have some coating of extracellular barrier?
No, you actually don't need, for these fish keratocytes, you don't need a specific extracellular matrix on that substrate. These are cells that are used to living on the outside of the cell, and so they don't depend on there being a certain ECM present. That's very different from most of the cells in our own body, which do require specific adhesions to specific ECM proteins. But that fish keratocyte doesn't. All right. So I want to focus in, and I'll be referring back to the actin cytoskeleton as I go through the examples of movements and mechanical interactions. And the actin cytoskeleton, I think, is a remarkable structure. It's kind of like Legos or Lincoln Logs. I guess those are quite old. In its ability to form a variety of different structures. You can find it in a huge range of different processes. I've talked about it involved in motility. But its ability to, a cell's ability to take up material through endocytosis or macropinocytosis, other processes for taking in material, all rely on actin. The same for getting rid of material or for releasing proteins, exocytosis. Movement of endosomes within cells also involve the assembly of these proteins. And more dramatic processes, like I was talking about for white blood cells, engulfing whole bacteria require this process. So these are kind of like the I-beams in this building. If you imagine this building was tired of being in the rain and wanted to move to, say, Berkeley, where it's nice and sunny right now, you could imagine disassembling all the I-beams and then somehow rebuilding them as the building moves towards the coast. That's kind of a complicated thing for us to do, but that's exactly what the cell is doing all the time. Its ability to translate from one location to the next means it has to take down its material in one place and reassemble it somewhere else. And that process of polymerization and depolymerization is central to the ability of actin to participate in these different processes. So actin, as I mentioned, forms different structures. I'm going to focus primarily on a branched actin network system. But it's worth noting that these same filaments can be organized in a range of different ways, from protruding bundles to stress fibers to cortical networks that are involved in cellular rigidity. Okay, so let's go back to this fish keratocyte cell, and told you a little bit about what those proteins are. The F-actin that's involved, filamentous actin that's involved in generating these protrusions, the vinculin involved in holding on to the substrate, and the DNA that is along for the ride. Okay, so these dendritic actin networks are called dendritic because of the highly branched structure. So what you'll hopefully see here is that we have individual filaments, and off of filaments are formed daughter filaments. And there's a specific branching protein that's very important in this process called ARCP3. So this branching protein will continue to form new filaments, and there's another protein involved that will eventually stop the growth of these filaments. And so you have this process of growing new filaments, stopping that filament, another new filament grows off of it. But there's one key additional ingredient, and that is you can only bind this branching protein if you're close to a membrane. And so that's why all of these branched actin networks grow near membranes, whether it's the leading edge of the cell, or it's at the membrane as it's undergoing phagocytosis, or other membrane-associated processes, because turning on that branching protein requires you to be near a surface. So this is a clever way for cells, or for biology in general, to be able to pattern assembly of structures. Rather than just having a soup of proteins that are mixing together and assembling wherever, by restricting one particular step of that process to a two-dimensional surface, that leads to the highly oriented branch structure that generates a force in one direction. So we'll talk about that in more detail a little bit later.
Are they disassembling at the back end, as it were?
That's right, that's right. So there's an assembly process and a disassembly process, and there are different sets of proteins that are involved in the assembly as well as the disassembly. And maybe an important thing to point out is that there's a link between these growing networks, which are generating a force as they add subunits to the end, and the adhesion sites, these little green things. At some point within the network, within the cytoskeleton, these force-generating structures have to couple to the substrate, or else you're just going to push the back of the cell out. So there's got to be some link between what's being pushed out and what's being held in place. And so that's the coupling between the force generation and the adhesion. Okay, so let's talk a little bit about, I've been talking about forces and generating forces and resisting forces and compliance. How do you actually measure these forces at the molecular scale? So there are a number of different ways, and all of them essentially rely on springs. So if you understand how a spring works, you pretty much understand how these sorts of force microscopy tools work. So with a spring, if you are compressing that spring, you can figure out how much compressional force is being generated. If you're pulling on that spring, you can figure out how much tension is generated. But you need to measure something. And typically, you need to measure two things, basically. You need to know what that spring constant is, and you need to know how far the spring moved. And so the techniques that are used for force microscopy differ quite a bit on what is the spring and how do you know how much it's moved. But in general, you're always thinking about those two things. What's my spring? How much is it moved? So two tools that we use in lab are one, optical tweezers, and two, atomic force microscopy. And I'll talk mostly today about atomic force microscopy because it has a number of applications in both cell biology as well as understanding molecular mechanics. In the case of optical tweezers, the spring is the potential, the gradient of the field created by focused light. And movement of a trapped particle within that field can be tracked by yet another field that allows you to quantify displacement on a very small length scale, a nanometer length scale. Now, the spring constants of these optical tweezers are quite small on the piconewton per nanometer scale. Atomic force microscopy has a range of different stiffnesses that are possible. Conventional cantilevers that are used or that are commercial are on the tens to hundreds of piconewtons per nanometer. So AFM. Now AFM, some of you may know, was really not developed for biological applications. It was developed for material science applications. The people who developed it wanted to be able to image the topography at the atomic scale, hence the name atomic, for non-conducting samples. So just before development of the AFM, the STM, the scanning tunneling microscope, was developed, and it relied on the fact that a tunneling current was passing between your probe and the surface you're interested in. And based on that tunneling current and a good feedback circuit, you can look at the topography of your sample. Now, that only works if your sample is conducting. So what if you want to look at a non-conducting sample? Well, that's where AFM came in. So AFM was originally just a conducting cantilever that had a sharp tip, and then an STM on top of it. So the STM idea was used to measure the distance, and then the spring was this little gold foil cantilever. And so now, with microfabrication tools, the cantilevers are now fabricated. They have tips with controlled sharpness, and rather than using an STM on top, we now bounce lasers off of them. So the current configuration that most people use for AFMs looks something like this. You've got a cantilever base that you can move in 3 dimensions. You've got a laser that's bouncing off of the end of your cantilever, and you've got some position-sensitive detector that allows you to sense what the displacement is, both how much it displaces vertically as well as laterally, if that's important for your measurement. Now, for biological applications, it was realized not long after development of AFM that you could immerse the system, and lasers will propagate just fine through most media, and as a result, you can do the same exact sorts of measurements to understand compliance of a material that you would want to do in air. You could also do them in liquid. And so a standard configuration for measuring mechanical properties of individual cells looks something like this. You've got a cell in the media, you've got the same cantilever, and you're bouncing a laser through that chamber. The kinds of information you can gather from AFM measurements are the traditional topography, the height information. Where is my cantilever for a particular force set point? But if I do something known as tapping mode, I can look at other features. I can look at the amplitude of the tapping signal or the phase of the tapping signal, and I can get different information about the interaction with that surface. So you saw in the last talk, the information that you're gathering was a potential. It was an electrical potential from a surface. Here, we're gathering very different information. Topography, stiffness, and I'll show you how much force is being generated by a particular cell. Now, the AFM by itself is a useful tool, but there's been a lot of interest in harnessing optical microscopy at the same time as force microscopy. So many of the AFMs that are now in use are in use as coupled instruments, where there's optical microscopy at the same time as AFM. And that simply means that while you're doing AFM, so the top is a phase image, and you can see fluorescent beads that are highlighted there. The bottom image shows some of the labeled proteins of those cells. You can combine the power of fluorescence microscopy, which allows specificity. You know which molecule you're looking at, and you can know how many there are, you can know where they move, etc. You can combine knowledge of the molecules with knowledge of the mechanics. So that's the motivation for combining AFM with optical microscopy. How many people have used AFMs here? A few people out? Okay, good. So there's some knowledge of AFM. All right, so perhaps the simplest experiment you can do with an AFM is to learn something about the stiffness of the material that you're looking at, or the cell that you're looking at, or about the adhesion properties of the cantilever with the surface, or if you've attached a cell to that cantilever, of the cell to another cell. So the ability to measure stiffness or measure adhesion forces simply involves moving your AFM up and down on a surface. And the surface of the cell is the sample of choice here. And what you're doing is you're saying, if I push and I displace my cantilever, I can measure that displacement, but I also know how much I moved down, so I know how much I've deformed my substrate, or my cell, and based on that, if I go over a range of different displacements, I can fit that to a curve and that curve fit, based on a certain type of theory, will get you an elastic property. So you can get a Young's modulus, essentially, out of a cell. Now, I should note a caveat here. I'm not a big fan of quantifying things in terms of Young's modulus, especially something as complex as a cell. Maybe an analogy is like if you were to crush your car, you could get a force displacement curve out of that. Here's how much force it took to compress my car. But it doesn't really tell you a lot about the car. And I feel like the same is true with some mechanical measurements of cells. Unless what you want to know is an overall bulk property, because that's important to the system, then single measurements on cells don't tell you a great deal. Now, I'm going to show you some single elasticity measurements on cells, because I think it is relevant for one of the examples I'll tell you about. But in general, I think it's worth thinking hard before doing measurements like this. Why is that data relevant? So this is the kind of traditional setup for doing AFM with cell biology. And I wanted to point out that there are a range of different ways that you can configure cantilevers in order to do more complex measurements. And so a few of the systems that we've developed in my lab involve using multiple cantilevers, or being able to work with non-adherent cells, or look at coupling. So let me tell you briefly what these are. The dual cantilever system uses two adjacent cantilevers to be able to normalize out for drift. Those of you who have worked with AFM know that drift is a real big pain. The cantilever base essentially moves relative to the substrate, and as a result, you don't really know where you are. You don't know what your reference point is for long timescale measurements. So to deal with that, we developed a system that uses one cantilever to figure out where you are, and a second cantilever to measure a sample. In cell biology measurements, you often want to know what's happening in the plane of movement. That is, if a cell is contracting against a force or protruding against the cantilever, you want to know what's happening in that plane. But most microscopes are set up to look at the bottom, which is not in the plane of force measurement. So we built a side view imaging system that lets us watch from the side as cells are contracting or protruding. In the case of non-adherent cells, pushing on a cell is like pushing on a crepe. It'll slide out from underneath the cantilever. And so to confine that, a simple hole in a substrate, microfabricated, was enough to solve that problem. Many times you want multiple points. You want to know what's happening at multiple points on a cell surface. And so combining AFM, either two cantilevers or one cantilever and a fluorescent probe, gives you information about propagation of stresses through a cell. Okay, so let me start with a couple of the examples I told you about. I'll start with one that is related to leukemia. So as a background bit of information, hematopoietic stem cells give rise to all the blood cells that we have in our bodies. Most people are familiar with the red blood cells and maybe the white blood cells, which are all these ones on the left. But there are also the platelets, as well as some immune cells that are involved in surveillance. So I'll be talking about problems that occur when the hematopoietic stem cells don't differentiate all the way to the end cells. So leukemia is simply a cancer of blood cell precursors. That is, leukemia occurs when the body doesn't generate enough of a desired cell. So if it realizes that you don't have enough neutrophils, it's going to keep cranking out the precursor cells for those neutrophils, but they don't divide properly, so you don't end up getting any neutrophils. You just get a bunch of these precursor cells that don't differentiate. And the same is true on the lymphoid side. So the result is this. So on the left is a normal, healthy blood smear. So if you take a drop of blood from your finger, you smear it on a slide, stain it, and look at it under a microscope, you see mostly red blood cells, these little things, and then a few white blood cells. That's about the right number. On the right-hand side is a leukemia patient. And so you can see many, many more blue cells, which are the white blood cells, as opposed to the red blood cells. And that's simply because the cells are not differentiating properly. They're not creating the right white blood cells. So what's the impact of this? Well, one of the ways that patients can die from leukemia is a process called leukostasis. And so leukostasis is essentially clogging of blood vessels. So these blood vessels are about the right size. You can see that they'll fit maybe one or two or a couple blood cells through them. This should be the same size as these vessels, but it has gotten much bigger. The reason it's gotten much bigger is because of clogging of this vessel. Now, what can give rise to clogging? There are a number of different ways that leukostasis could occur, and we wanted to explore the possibility that limited cell deformability was a problem. And so this is one example where measuring an average modulus or average elasticity for a cell might be a useful thing. If white blood cells have to pass through small vessels and they have to deform when they do that, then being able to deform is a very important part of that white blood cell. And if they can't deform, just as trying to push something through a small tube, if you can't deform, then you're more likely to get clogged. So we wanted to explore whether that was the case. If we look at leukemia cells versus healthy cells and look at their mechanical properties, is there a difference? What we found was, in fact, there was, if you look at patient samples that are symptomatic for leukostasis, the average stiffness is greater than those who have leukemia but do not have signs of leukostasis. And so you can look at the difference. There's quite a significant difference. And that's just taking average measurements of stiffness of these patient samples. Now, perhaps more interesting is how you treat leukemia and what effect that has on stiffness. So the standard way of treating leukemia is chemotherapy. So you give a drug that essentially kills white blood cells. Kills many cells, but in particular, white blood cells. Now, what happened when we exposed myeloid cells, in particular, to a chemotherapeutic, daughterubicin in this particular case, we found that the stiffness of those cells shot up over time, while for control cells, nothing happened. So simply the fact of giving leukemia to patients causes a stiffening of those leukemic cells. And it progressed even before apoptotic markers, which would say, okay, this cell is dying, it's in the process of dying and going to be removed from the bloodstream. The stiffness increased. And so what's responsible for that stiffness increase? What we found was if we use a drug that disassembles the actin cytoskeleton, we're going back to who's the culprit here? What is responsible for this change in stiffness? If we look at the actin cytoskeleton, that it seems to be a major part of this stiffness increase. So if we disable the actin cytoskeleton, then the stiffness change is not as pronounced. And so what this suggests is that the stiffening of white blood cells during, both before and during treatment for leukemia may be a causative factor of clogging of the vessels, which can result in rupture and then death. And so this is one example of where mechanics plays a central role in potentially in a clinical disease. But AFMs are not convenient things to have in a hospital. You're not going to probably find any hospitals that routinely will measure samples with an AFM. So there's got to be a better way of quantifying stiffness. And so we set out to try at least one way, and microfluidics offers some possibilities. And so we simply built a system in which we could look at the transit time of different cell populations. And so using a very simple flow system with channels that were fabricated to emulate the smallest capillaries that these blood cells would go through, we found that we could quantify the time it took to transit across a smaller chamber, and we could use that time as a measure for the stiffness. So an example, if we took a normal neutrophil and we pass it through one of these chambers, that's the same neutrophil going through, not different neutrophils. You can see that it moves relatively quickly. Now compare that to a neutrophil that's been activated. And so it's been activated with the same chemical that caused the neutrophils to come out of the blood vessels to the puncture wound in the fish, FMLP, and that activates them, stiffens them, and you'll see the same channel, it's much slower to go through. And that simply has to do, there's no adhesion, there's no coating on this channel. It's simply flowing the cells through. You can see that there's a difference in the time scale. Now, time scale and stiffness correlate. They're not perfect, but they provide some indication of which cells may be most prone to aggregation and to leukostasis. And so here's an example of patient samples as well as some control samples, and transit time being much longer. So this issue is not just confined to leukemia. There are a number of other diseases in which blood cells or other cells are affected, and stiffness may be playing a role in transit. Okay, so I want to go to the second example, and it's also going to be a blood cell, but I want to show you a couple movies before that. And that has to do with forces generated by individual cells. So take platelets, for example. Platelets are able to generate forces during clots. And most cells in your body are actually able to generate forces, contractile forces, because they're not going anywhere. Only a few cells in the body are really motile. The rest are involved in a contraction. So I'm going to show you a movie of mammary epithelial cells. So these are epithelial cells that are charged with growing a branched network. And what you'll see is in this gel, there are a set of different particles. And the different particles are, you can see the cells growing, but the particles are also being pulled inwards. And this ability to contract is also thanks to the cytoskeleton. So the cytoskeleton combination of actin and a molecular motor called myosin are able to pull on adhesions and therefore deform substrates. So here you're seeing a combination of motility as well as contraction of a growing tissue. So can we measure these kinds of contraction forces with existing force microscopy tools? The answer is yes. AFM and related techniques are able to quantify the scale of forces that these types of cells are able to generate. And the method for doing that is simply contraction between two surfaces. So just as the cells are pulling on different adhesion sites, the ability to mount the cell on one surface and a second surface and watch as the cells contract, we can quantify again with this spring. If we know the displacement, we can know the force. We simply allow the cells to flow into a chamber. As they touch on one surface, we move the cantilever over and lower it down onto the cell. The cell will then spread on both surfaces if they're coated with ECM. So this is an example where the extracellular matrix for these epithelial cells is important. And so if we coat both the bottom surface and the top surface with the same proteins, and we move the cantilever on top of the cell, it will spread onto both surfaces and then pull. Okay, so here's a side view image of what it looks like for this osteosarcoma cell as it pulls on a cantilever. So the cantilever is on the top. There's a substrate right here, and you can see a bit of a reflection of the fluorescence below. So you're looking at the actin inside of this cell as it pulls on these two surfaces. As you can see that over this about 5-minute movie, it's quite a dynamic rearrangement of the actin cytoskeleton as it's pulling on this structure. So being able to quantify things about the forces that are generated allows us to say something about how much shape change, quantify forces and shape change, allows us to say something about how the cell responds to its physical microenvironment. So we can measure things like the cell length, how much force it's generating, and how does it change shape over time. So this is, again, just using AFM, but in a slightly different configuration that allows you to isolate specific quantities. So now let's get to platelets. So does anybody know how platelets are formed? They're actually formed in a really cool way. So they're formed, I'll show you this movie, from megakaryocytes, which are the precursor cell. And the megakaryocyte basically fractures. It beads off into lots of smaller cells, and those individual parts of the cytoplasm are the things that we call platelets. And so one could argue about whether platelets are really cells or just fragments of cells. So that process of fragmentation, all those little individual particles, those are the platelets. And so what those platelets are able to do are critically important at all times, essentially, not just when we have a big wound, but when we have small ruptures in our vasculature over time. Those small ruptures are constantly being repaired and healed and covered by platelets. So what's the process? So blood clot formation, you have exposure of collagen due to a rupture in a vessel, could be a very small one. Fibrin is polymerized and the clots, the platelets grab onto that fibrin gel and pull it together so as to seal up this wound, close the hole. So that clot contraction process has been studied for years, and a bulk assay for doing that is simply take some platelets, mix them with some fibrinogen, and watch them contract. So this is a very simple experiment to do. You can do it yourselves, take a little drop of blood, and isolate the platelets and see what happens. So I think this is probably my blood. I think that these are the experiments that I think the grad students enjoy particularly when they need to harvest some blood from their advisor. And you can see over the course of a couple hours, the gel contracts. So it undergoes this dramatic contraction. And the thing that's doing that contraction is this little particle that came from the megakaryocyte, the platelet. Okay, so it's able to undergo this dramatic change, and it's able to do that at the same time as it's changing the physical properties of its environment. So clot stiffness is an important property in both the ability of a platelet to close the wound, but it's also in a number of clinical conditions. If you have a soft clot, it's more likely to break off and move through the bloodstream and potentially cause a stroke. So having clot integrity is an important thing. And the ability of these platelets to not just pull things together, but pull things together and change the mechanical properties of the clot is one of their key attributes. Now, the studies that have been done so far are bulk studies. There haven't been studies on individual platelets and how much can an individual platelet pull. So we decided to try that same configuration of placing a platelet between a cantilever and a surface and ask, can we quantify how much force is being generated by an individual platelet? Part of the reason why it hasn't been done is it's hard to catch them before they activate. They're really, really ready to go. As soon as they sense a fibrinogen-coated surface, they're going to activate, spread, and try and pull. So it's very difficult to catch them in the act, essentially. So the way we did that was by watching them as they were floating around in solution from the side, using the side view AFM. And then as soon as they came close to the surface, we moved on top of them with an AFM cantilever and held them in place and watched as they contract. Yep.
So given that it's got a solid surface at the bottom and the AFM cantilever, both of which are likely to be stiffer than its usual environment, how similar is the force that you're calculating here to what is really exerted in vivo situations?
So these are going to be maximum forces. So we're actually going to get to exactly that question, which is how do you best emulate the microenvironment? So one way to think about it is that the flexibility of that cantilever is probably the best measure of what the platelet thinks it's pulling against. So in this diagram, it's pulling between two fibers, and in this diagram, it's pulling between the cantilever and the surface. So essentially, the stiffness of the cantilever is the thing that is emulating the microenvironment. And your question, I think, is that really a good measure of what's going on? So actually, in the case of the cantilevers that we used, it's a reasonable estimate for the stiffness of a clot. But I'll show you the two extremes. That is when you prevent it from deflecting at all, which is a very, very stiff environment, or when you make it even softer, what happens. And you'll see that there's an interesting capability of the platelet to sense that environment. Okay, so in these experiments, we look at force generation. It's able to contract. It's able to contract with surprising amount of force. So just to put this in perspective, this is probably about 20 times stronger on a per volume basis than almost any other cell. So these platelets have almost nothing in them but the machinery to do contraction. They release a number of factors to stimulate more coagulation, but they're highly tuned to do contraction.
Machinery to do the contraction, you mean actin-myosin?
Yep, actin-myosin contraction, exactly. Yep. So again, it's the cytoskeleton, but it's only the bits of the cytoskeleton that do contraction that are involved in this process. So if you disable either of those, it doesn't work.
Does the adhesion to the two surfaces enter in at all in terms of how well it's adhering? And then
It does. So the adhesion to this fibrin network is essential. Otherwise, it's going to, if that's the weakest link, then it's going to pull itself off of these fibers. And so as we conducted these experiments, at the end of them, we can do an experiment in which we pull against just increase the force until there is rupture. And that force was always substantially greater than the average contraction force we were able to get.
so there's no variability that you know of in what you're measuring there in terms of the adhesion between the two surfaces. So that's kind of a fixed thing.
Well, it's going to depend a lot on the concentration of the fibrinogen at the surface, or sorry, not fibrinogen, the polymer at the surface. If that coating is non-uniform, that could affect the overall adhesion. But the individual adhesion strength has been measured, not in our experiments, but in other experiments, and it's the number of receptors times that receptor ligand adhesion strength is always much greater than the contraction force.
I'm sorry, one more technical question.
Sure.
Integrin, coherent, mix, what?
So primarily integrin.
Thanks.
Okay, so this contraction takes place, and you'll notice it takes place over a relatively short time scale. And so this is a cell that is built to contract and does it once. Once it contracts, it's done. So if we allow the force to, allow it to contract, and then we try to release the force. So using an AFM, you have the ability to change the load that's on your sample. And so if you reduce the force on that sample, one might think that it's only just stalled here because it's not strong enough. But if you reduce that force, it doesn't do anything. It doesn't contract anymore. It's done. So it's an interesting structure in that it's a one-shot contraction machine. Now to the question of the microenvironment. Is the stiffness really an important factor? What we found was that if we change the stiffness of the cantilever, that is, we use different stiffness cantilevers, or if we do a case in which we feedback such that the separation between the cantilever and the surface can't change, that the stall force and also the loading rate, the rate at which the force increases, changes. But what that says is that the platelet machinery is sensitive to the stiffness of the clot, and in fact is able to pull harder on a stiffer clot. Now why might we want it to do that? What would be a good thing about pulling harder on a stiffer clot? Well, there'd be one other piece. So I don't know the answer, but I'll give you a hypothesis that we have. And that is, if you have a heterogeneous clot, which is most likely, there's probably not a great way of creating a clot that is completely uniform in its density of fibrin or its density of platelets. So you're bound to have different amounts of contraction and different mechanical properties. If you have a platelet that can pull harder on a stiffer clot, you have a greater chance of uniform contraction of that clot despite it being heterogeneous. So that's our thinking for why this might be of interest. So we were able to measure the elasticity in this configuration, about 10 kilopascals, and extensibility. So even though it's contracted, it's able to, it's extensible. It can be stretched without breaking. Okay, so back to this question of why might it be stiffness dependent. So here's the hypothesis. If there is heterogeneity in this clot, if you're going to have a contractile element that's sensitive, I'll pull a little bit. If it's soft, I'll pull a lot. If it's hard, then you get a more uniform contraction of the clot. So we're working on ways to test that very idea. Okay, so the question on microenvironments is exactly one that we've been wrestling with. So AFM is great, but the cantilever stiffness is the cantilever stiffness. If we wanted to change the apparent environment of the platelet or of any other cell, we had to change the cantilever. And that's really a slow way to do work, and that's a kind of difficult, if you want to really understand the way that cells react to changes in their physical environment, or really how the mechanics can regulate the behavior of the cell. And so can we vary stiffness dynamically? And so we thought about this for a little while and decided that with the appropriate feedback mechanism, we ought to be able to do that. So the idea is this. I can pretend that the cantilever is a stiff spring if, as the cell is pulling, I measure how much it's pulling, and I displace the base of the cantilever up by a certain amount. So I can essentially fight the cantilever fight the platelet as it's contracting. From the platelet's perspective, all it knows is that it's hard to pull. And if it's hard to pull, that's like a stiff surface. So it's very much like if you catch a baseball. If you don't give any with your hand, from the baseball's perspective, that's a really hard surface. It's going to hit you and then fall off. If, on the other hand, you move your hand with the ball, from the perspective of the baseball, that's a really soft surface. That seems like, my displacement and my change in force the slope was low. So that's the basic idea here, is can we move the cantilever as a function of how much force is being generated to create different apparent stiffnesses. And so we worked out how to do that. The basic idea is that, as I mentioned, to make something appear stiff, move away as it's pulling, and to make it appear soft, move with it as it's pulling. And then you've got the ability to vary your stiffness over a wide range. And interestingly enough, force clamps, which are routinely done in AFM and optical trap and other type measurements, force clamps are just an extreme in one direction. A force clamp is a very, very soft surface because you have no way of increasing force. You can displace all you want. You don't increase force. And a position clamp, you can pull all you want. You don't change position. That's an infinitely stiff surface. So you can get a full range of stiffnesses from a single cantilever. So we demonstrated that with some control experiments, just using an expanding gel, using a feedback algorithm that adjusted the movement of the cantilever base. We could see that the slopes were different, and these different slopes are different apparent stiffnesses. So what about with a cell? What you can do now that you have an ability to change stiffness dynamically is you can ask, does a cell sense that rapid change in stiffness? So if I've got an environment, if my clot is changing its stiffness rapidly, do I have some way of sensing that? Or is one platelet going to sense one stiffness, and only when it's done and another platelet comes in, can it sense that next stiffness? So we wanted to test that. We test that with initially with fibroblast cells. And what I'm showing you in blue is the step change in stiffness. So these are the stiffnesses that are programmed in to the feedback routine. We're going from 1 times K, which is the cantilever stiffness, to a fifth of that, 0.2, to 5, to 1K, and so on. So we're stepping among different resistances to contraction. And what you notice is that there's a marked change in slope each time we change the apparent stiffness. So we're not changing the length of the cell at that instant. We're not changing the force at that instant. The only thing we're changing is the stiffness. And if you do this, if you zoom in, and if you do this a number of other times at different frequencies, we found that the contraction of these fibroblasts, at least, is sufficient is sufficiently fast to respond to changes in stiffness that are under a second. So half second, even smaller. So very rapid changes, very rapid sensing of stiffness. Not of force, not of length, but specifically stiffness. Yep.
So I have a question about this, in the sense that here you're having a stiffness change that is only in one direction. That is only the cantilever on top is changing in stiffness. Whereas put the cell in a platelet, the stiffness is going to be altered in different directions. So is there any way you can, say, soften the surface on which it is or something and see if that kind of alters the way, the dynamic of its...
That's a great suggestion. So that's exactly right. We have one dimension has a certain stiffness, and then the other dimensions has a different stiffness. So it's stiff, the cantilever and the substrate are both stiff compared to the change in stiffness in one dimension. I think these experiments show two things. One is that changing the stiffness in one of those directions is sufficient to elicit a response. So it doesn't just listen to the highest stiffness direction, it actually is watching for how stiffness is changing in multiple directions. Second is I think we can address that question you have by changing the substrate, making that a gel rather than a solid substrate, and asking if there's a third stiffness, how does this behavior change? We haven't tried that experiment, but that's a good idea. Okay, so for the last example, I want to go to the actin networks. So we've been talking about whole cells, first white blood cells, and their overall stiffness. We talked about platelets and their ability to contract and how the physical environment adjusts how they respond. I want to go now to specific molecules within the cells that are responsible. Cytoskeletal molecules involved in protrusion of crawling cells. So here's that movie again of it running into the surface and responding. And the question, what's going on at that very leading edge that allows it to know something about the force? If this is really just first order reactions like you learn in chemistry, there's no force term there. There's no way for force to be coupled in to the biochemistry. So how really does that happen? So this is the network. I explained it briefly. Filaments polymerize, they branch, and they get capped, and you have to be near a surface in order for branching to take place. And that's how you get these organized, oriented networks that are generating protrusion. Okay, so how do you convert force into biochemistry? And this is one of the key ideas in mechanotransduction. That there's some way that you can go from physics, which we learned, to biology and biochemistry that we're learning. There have been a number of ways in which this seems to be happening. One is at adhesion sites. So adhesion sites can be sensitive to the stresses that are applied to them, either by changing the overall structure or increasing the number of proteins that are present. Early on, it was known that motor molecules will slow down their enzymatic activity, will slow down their walking with force. And in particular, this was for muscle myosin. So you apply load to a muscle. If you try and lift weights, and you try and lift them really fast, there's going to be a force-velocity relationship for that. Finally, unfolding. If you unfold a molecule, you may be exposing new places for molecules to bind, and that can change the activity of the system. So the problem is that at the leading edge of the cell, none of these are really at work. The first adhesions in crawling cells like this don't occur for until a micron or so back. And so force could be coupled into those adhesions micron back, but at the very leading edge, it's just the network of branched filaments that are present. So is there anything inherent in those that could be sensitive to forces? That's what we want to understand. Okay, so again, we'll use our old friend, the AFM, and see if we can figure out a way of quantifying forces and properties of these networks specifically. So in order to isolate what's due to a focal adhesion, what's due to protein unfolding, what we really need to do is take out those proteins of interest and put them together isolated from the cell. So in experiments that I'll show you now, we're doing reconstituted experiments. So these reconstitutions are a process of identifying the particular proteins that are important for a process, purifying them, and then recombining them in a physiologically relevant way. And so the way we've tried to emulate what's going on at the leading edge of a cell is to have soluble proteins in solution here. So the actin, the branching protein, the capping protein, cycling proteins in solution here. And then to have on the cantilever surface, to have this protein that restricts the binding of the branching only to near the surface. So we only have one control, one thing that keeps this from being a random polymerizing network everywhere, and that is putting what's known as a nucleation promoting factor on the surface. And that's just like what the cell does. It keeps that nucleation promoting factor on the cell surface. Okay, so we do AFM experiments with this purified system. What you're looking at in the bright channel there is labeled actin. So as the actin assembles in this network, it's assembling at the cantilever surface, you can see that it pushes the surface down. Or in this case, we have a force clamp in place, so we're not letting the force increase, and we're simply allowing the network to displace at a constant force. And so what this is showing is that with a few proteins mixed together, you can recapitulate the growth of an actin network. Not only that, but you can show that it generates force. It's generating a controlled force underneath this cantilever. So now that we have it in a force microscope, we can do a range of experiments with it. We can look at its elasticity, and we did this a couple years ago to show that the elasticity is quite complex. There's a nonlinearity to both the stress stiffening, and there's a softening behavior that we attributed to buckling of individual filaments. So the elasticity of these networks is unique, and the buckling process results in a reversible stress stiffening. That is reversible in which if you let the stress off, the network recovers its elasticity, and you can go right back to its original elasticity. So other than the mechanical properties, it's really the dynamic properties I want to tell you about. So a way to characterize how dynamic networks work, or how motor molecules work, is to measure a force velocity. That's kind of like, for your car, it's like a torque RPM curve. You want to know how many RPM do I need to go to in order to get a certain torque. Well, it's similar here. How much force can be resisted for a given growth rate. And so to measure that, you can do that easily with a force microscope like this. Simply allow the network to grow. You're measuring rate of displacement, and you're also measuring deflection, which gives you force. So you've got force and velocity directly from a growth curve. And so if you take the growth curve, it looks something like this, and you plot it as a function of velocity and force. What this is saying is that at some point, if I push hard enough, I can stall the growth. This network can't keep assembling if I push hard enough. But if I don't push quite as hard, I'll go faster. And over some range, that's about half of this force-velocity relationship, I get a constant growth rate. That is, I'm growing at a constant growth rate. I'm adapting to this force. I'm not slowing down. So what could be responsible for that? Well, our hypothesis is that what's happening at these branched actin networks is that the application of a force is changing the density of the network. And if I nucleate new filaments, if I create new pushing proteins, what I'm doing is I'm spreading the load over more force-generating polymers, and I'm able to handle a greater load. So that's the idea, that as I increase load at the edge of this growing network, I'm actually nucleating new filaments. Okay, so we've recently seen through a range of experiments that the curvature of the filament affects the branching behavior, and this curvature leads us to believe that when these networks are under load, they're more likely to branch on a surface that generates a new filament towards that surface. So I'll give you a diagram here. So here's the data. If I've got a filament and it's bent that way, I see a greater chance of forming a branch on the outside than on the inside. So this is a clever mechanism for a single filament to be a sensing element, to be a force sensing element, and to bias the growth of new filaments in a certain direction. Okay, so I think I've got to finish up here. So here's the model for what we think is going on. as the filaments are being deflected by a surface, whether it's a cell that's crawling into a surface or whether it's a cantilever surface, filament bending is actually coupled to the branching process, and that coupling leads it to generate more branches in the direction of the load. So again, the idea that force feedback resides within these proteins, within these cytoskeletal proteins, doesn't require the nucleus, doesn't require you to ask questions of the nucleus or transcribe more proteins. This ability to sense and respond is all resident in a small set of proteins. Okay, so I think I will end with one more movie, and that is, does this make any sense? Well, we can do a quick experiment to see if the presence of a force changes the amount of actin that's present at the leading edge. And so this is one of those experiments where I thought, it's not even worth doing. We shouldn't try it, but the students did anyway, and it did work. So what I'm showing here is one of these fish keratocyte cells, and it's running into a glass pipette that's put in front of it. So if we want to know what happens when cells run into objects, what the response of the cytoskeleton is, the simplest way is to transiently put an object there and then figure out how much actin is there. In order to do that, in order for the disassembly process not to take over, we need to freeze the actin, we need to freeze the cell instantaneously. So what you're seeing, that little white flash, is a fixative, glutaraldehyde, that gets washed over the cells and suddenly freezes them in place. Okay, so that's the process. They run into this glass pipette, and then they're frozen in place. And if we then do a phalloidin labeling of the actin, you can see that there's this line of increased density at the leading edge. So it seems like the idea that increased density occurs due to mechanical interactions seems to be true in at least these cells. So I think I'll end there and be happy to answer any questions.